Hello, hello. Welcome to XC Psychology. Today, we're going to talk about ion channels. And as you can see from the screen, there are many channels, and we have talked about our familiar friends here. A lot of ions, potassium, sodium, calcium, chloride, and they move in and out of the membrane like the burger, in and out. This episode is sponsored by In and Out, and definitely a burger fan, but that was a wishful thinking. In and Out, if you are watching this video, please considering sponsor me. So before we talk about ion channels, let's first talk about what are ions. And we've actually been talking about ions a lot in our previous videos about potassium, sodium, the salty banana, and chloride and calcium. So here is a official definition of ions. So basically, it's an atom or molecule that has net electrical charge. That's the key phrase. Net electrical charge that possesses an equal number of protons and electrons. I know it sounds very confusing. Before we look at this, table, let's first maybe look at some practical examples. So basically, we have atom. Atom is very famous for wanting to keep a balance. And it has protons, which are usually positive charged, and electrons, which are usually negative charged. And let's take sodium for an example. So if we have 11 protons and 11 electrons, it's a neutral sodium, and the net charge is 0. And imagine we've lost an electron, and now we have one more positive, right? So the net is positive 1. And that's how we get our sodium ion. And so atom loses electrons. This has made our sodium ions a cation. So see, cation. And cat is so cute, so it's usually positive. So let's have an example about negative. If we have 17 protons and electrons, this is our neutral chloride. It doesn't have, it's equal number of positive and negative. But what if we have gained another electron? Now it becomes 18. And the net charge is currently -1. We have our chloride ions. So see, that's met the definition here. We have a net electrical charge, positive or negative, and we have an equal number of protons and electrons. And so gains electrons now makes the ion anion, and the chloride ion is anion, it is negative. Now let's look at this table. It's a very good summary. Basically, we have cation, positive, and anion, negative, and electron count. If we look at our example here, it's definitely for cation, it's fewer electrons than protons, so it's positive. And for anion, it's more electrons than protons, so it's negative. How they form? Atom loses one or more electrons, so they become positive. And how they form for anion gains one or more electrons, so it becomes negative. And cation is usually formed by metals such as sodium, potassium, calcium. And anion usually is non-metals, so it's chloride and oxygen. So to summarize here, you remember our old friend back when we were studying action potentials and voltage change over time. We have our sodium with a cation positive. It is used for depolarization during the action potential. We have our potassium Remember salty banana, which is also a cation. It helps with repolarize the neuron after actin potential by leaving the inside of the neuron through the membrane. And we have calcium. Remember, we bind with vesicles and makes the vesicles drop down and fuse with the membranes and release the neurotransmitters. And we have our chloride, which is an ion, usually makes the neurons more negative. If you remember our last episode about IPSP. So now we've understood what are ions. Now we are trying to answer the question, what are ion channels? And basically, they're just proteins. They're proteins that forming pores along the membrane of the neuron. And if you don't understand what a pore is, we've talked about gates. So it's kind of like a gate, or some people say it's a hole. That allows ions to travel in and out of the neurons. And why do we need ion channels? Very good question. So first, before we look at all of this, let's look at a membrane in detail. So our membrane actually is a very interesting design. So outside the membrane or outside the neuron, we have this extracellular fluid, but we short as ECF. And you remember our, players here. We have sodium, we have calcium, we have chloride outside. And the extracellular fluid actually accounts for 1/3 of our body water, right? And inside the membrane, we have cytoplasm. Basically, it refers to the gel-like fluid that fills the inside of a cell enclosed by the cell membrane. Basically, everything inside the cell membrane, we can call it cytoplasm as an umbrella term. And what is here? Here is what we call a lipid bilayer. The bilayer means two, right? It has 1 and 2. And the bilayer is a very interesting structure. It has a head that is this little blue circle here, is hydrophilic, means water-loving. It loves water. I remember the outside of the membrane, extracellular fluid, is mainly, water. And it has tail. It's called hydrophobic. So the tail is hydrophobic, means water fear. It doesn't love water. It's kind of like water and oil. And so why is this design makes it tricky for ions to travel or communicate or move? So because outside is mainly water, if you look, one single ion, like say sodium in detail, it is usually attached with water molecules. Same with chloride and calcium. So just imagine those little ions floating around in extracellular fluid here actually are attached with water. Inside, same, we have our negative protein, right? And we have our potassium. And potassium is also attached with water. Remember, it's gel-like, fluid. Fluid means water. And head, they love water. And so if water and oil doesn't, get along, how do we help them communicate, right? And hence we have this design of, we'll just use sodium channel as an example. We have a design of the sodium channel. This is a sodium channel that is ready, right? Basically, what you see here is a special tunnel that allows sodiums to travel in. And a fun fact for this is that when the sodium, right, if you look here, when the sodium travels in, it actually, during the process, let's just put this one here, the gate will say, do you have water? And sodium says, yes, I do have water. And the gate will say that you can go, but the water needs to stay. And so it removes all of this water here and just let this sodium ion passes through, right? It's the same for potassiums to leave the channel. It will also ask the potassium, hey, do you have water? And then, yes, I do. And then it will just only allow the potassium to go in and strip off the water. And once, say, they've passed the membrane, whether in or out, they will be reattached with water. And I think that was a super fun thing to learn about our system. And if you want a good summary, here is a table. Sodium outside is high, right? Inside is low. Potassium inside is low, outside is high. Chloride outside is high, inside is low. And proteins outside is low, inside is high. Because of the negative proteins, we have a resting potential of negative 70 millivolts. So just to have a quick overview here, when we talk about membrane, the membrane is actually connected and wrapped up with the neurons, right? So when we look at it, is extracellular fluid, inside is cytoplasm, and outside is extracellular fluid again. Just in case when we're looking at the details, it may mislead you to think otherwise. So now we're looking at the types of ion channels. And in previous episodes, we have talked excessively about voltage-gated channels. And their function is merely generate and then propagate electrical signals or action potentials. We've talked about the wave many times. And in recent videos, we learned about ligand-gated channels, which are very selective for, certain neurotransmitters to bind to. And for this episode, just simply remember it as fast communication between neurons at synapse. And we have some other types of ion channels as well. One thing we need to know is the leak channels. Remember when we talked about action potentials and the voltage-gated channels, and somehow it will hyperpolarize. And because gates sometimes leak, and we do have channels or gates that are leaking all the time, this is to maintain the resting membrane potential. If everything is airtight, I think there's extra work for our bodies to do to maintain that resting potential. With leakage channels, our body or our nervous system will find a way to rebalance itself by moving in and out the right ions. And some other things, mechanically gated channels. It will detect the physical forces such as touch, pressure, or stretch, or hearing. Remember, our brain relies on the neural signals to sense, perceive, move. So definitely we have mechanically gated channels. And we also have temperature gated. We will detect the heat and cold. And here is a fun fact in very small font. Remember in the last episode, we talked about this little guy here, axon initial segment. It has extremely high density of voltage-gated sodium channels. And I think you know why. And so what makes the ions move in ion channels? If you look at this little penguin here, I'm sorry, the answer is not pump. I repeat, ion channels do not pump ions. Ion channels do not pump ions, right? So the movement is actually very, very passive. This is the key word here. It's driven by the ion's electrical chemical. Remember when we talk about the synapse transmission, synaptic transmission, we will talk about electrical, chemical, electrical. And so it makes sense here is electrical, chemical gradient that combines concentration gradients, which simply means ions move from high concentration to low concentration. And electrical gradient, which means positive ions are attracted to negative ions, and vice versa. And if you are confused by this super fancy formal terms, just remember when we talked about action potentials and how we open the gate, it's usually start with the binding of a neurotransmitter and then becomes very positive inside the neuron. And remember concentration gradient, we move from high concentration to low concentration. So if here we have a high concentration of electrical charges or ions, you'll want to move to the low concentration here. And initially, as we remember, the membrane resting membrane potential is negative 70 millivolts, and it's way more negative than the outside. And so electrical gradient, same thing. This one has become more positive. It will want to go to the negative regions like a wave after wave after wave and after wave, and opening the sodium channels here, 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 and here. And we already know it. It's just now we are putting them into a formal terms: concentration gradients, electrical chemical gradient. And just remember, for the ion channels, it is passive. Why is passive? If you still don't think it's passive, you still feel to struggle with this concept, just remember it needs the positive charge for the gate to open. It's not like something pushed the door open. It's more like, hey, oh, wow, here is positive charges. I'm going to open the gate. And which brings us to what's coming up next. What's coming up next is ion pumps. Ion pumps is another way to move ions, and it requires a lot of energy, like ATP. Ion channels, they don't need ATP, they don't need energy. And this one is a lot more active, not a passive way. And this ion pump, and together with our ion channels, together they form the ionic homeostasis, enable the electrical signaling in our neurons. And hopefully, in the future episode, we can specifically talk about ligand-gated channels and neuroreceptors. That's it for today's video. I will see you next time.